Hello everyone, and welcome to the first lecture of Unit 2. In this lecture, we will go over some foundational topics for microbial genetics and talk about the process of taking the information in genetic material and turning it into proteins. There are 3 lectures overall for the microbial genetics chapter in your reading, so this is the first one of them. Make sure that you watch the other 2 after this one as well, or you will be missing information. As always, you have your lesson objectives here. Hopefully by now you have an idea of how you like to use them, how they work for you, but these are just the main points that you should know after working through the reading and listening to this lecture. So look at them beforehand to make sure you know what you're listening for, and then review them after as well to make sure you hit the key points. On these next several slides, we will go over some background information, some foundational information that you will need to understand the rest of the topics in the microbial genetics chapter and the other 2 lectures. If you took your introductory biology course recently, this will probably seem like a lot of review material. If you took it a while ago, hopefully this will help jog some memories and bring some information up to the forefront of your mind that you may not have thought about in a while. But either way, make sure you pay attention carefully to these next couple of slides so that you're clear on all of the concepts that you need to understand, the vocabulary terms, etc., because it really does lay the foundation for understanding the rest of the material in this chapter, and to a certain extent, the rest of the material in this unit and parts of units 3 and 4. To start us off, I want to review some vocabulary terms and some pretty broad topics that are important when we're talking about DNA and proteins and passing information from one generation to the next. So DNA is the molecule that is contained within organisms that has 2 main functions. The first main function is to allow the organism to create proteins. So the code in the DNA specifies what proteins an organism or a cell can make and when it makes them. And then the other main role that DNA plays in biological organisms is passing information from one generation to the next. So when we talk about DNA in the context of genes and creating proteins and using the information contained in those genes to create proteins, we're typically talking about genetics, or we use that term to describe that context of DNA. And then heredity is the aspect of DNA where the information is passed from one generation to another. Some other key terms are genotype and phenotype. So genotype is the genetic makeup of the organism. It is literally just the actual sequence of A's, G's, C's, and T's that are contained in its DNA molecule. The genotype of an organism, basically the order of the nucleotides in that organism, determines which proteins the organism can make. It can help determine when the organism makes those proteins. And for eukaryotes, the genotype is fairly stable, meaning that most of the time throughout a eukaryote's lifespan, it doesn't change very often. For eukaryotes, what they are born with in terms of genotype is typically what they die with. There's not much change over the life of the organism. So if an organism has a certain order that says AGCT at the beginning of its life, that's what it's going to carry out through its entire life, and pretty much that's what it's going to die with. However, in prokaryotes, the genotype can be quite variable, actually, because if you think back to Unit 1, we talked about plasmids and how some prokaryotes, and sometimes many prokaryotes, will acquire plasmids throughout their lifetime. So the genotype in prokaryotes can change fairly distinctly or fairly significantly throughout the prokaryote's lifetime, in part due to plasmids and in part due to other events that we call genetic recombination events. We will talk about genetic recombination events in another lecture, But for now, the big thing that you want to know about genotype is that genotype is the genetic makeup. When we're talking about genotype, we're literally talking about the order of the nucleotides, the order of the A's, G's, C's, and T's in an organism's DNA. And that for eukaryotes, the genotype is fairly stable throughout its lifetime. But in prokaryotes, it can be fairly variable. Now, the next term is phenotype. And a lot of times people have a very difficult time distinguishing between what is phenotype and what is genotype. But phenotype are basically the physical and metabolic characteristics of the organism that result from the expression of the genotype. What that means is the phenotype are the physical or metabolic traits that an organism has, basically the proteins that it contains, as well as other characteristics like its shape, etc., that are a result of the proteins that have been created from the genotype. So genotype is just the order of nucleotides. That's what we're talking about when we're talking about genotype. And the phenotype is the result of the expression of the genotype, which again just means the proteins that were created from the code in the DNA. For both eukaryotes and prokaryotes, but especially prokaryotes, the phenotype is highly influenced by environmental factors. What that means is that the external factors for that organism, what type of environment it's in, maybe how much light it has, what types of nutrient sources it has, etc., can all affect which parts of the genotype are used to make proteins, which will affect the phenotype, right? Because the phenotype is just the expression of the genotype. So let's say an organism can produce a pink protein or a yellow protein. And if it has a lot of light, it produces a pink protein, but if it doesn't have very much light, it produces a yellow one. If we have an organism that is exposed to a lot of light, that will switch on the genes to produce the pink protein, and that organism will produce that pink protein and may like pink as a result. However, if it doesn't have a lot of light, then that will cause the genes, that part of the genotype, for the yellow protein to be expressed. Basically, that protein will be made, and that organism may look yellow. So the big overall concept with that is that the phenotype, how an organism looks, or what types of metabolism it is creating at that moment in time, is, or at least can be, very dependent upon environmental factors. The phenotype also, for both prokaryotes and eukaryotes, may change, even significantly, over the organism's lifetime. Again, going back to that example I just had, that the different environmental conditions of our organism that we were talking about with the pink protein and the yellow protein, the different environmental conditions may cause the differential expression of the genotype, and as a result, cause a change in the phenotype of the organism. Now, looking at this image, you have what we call just a classic Punnett square with a cross between two parents, and this code up here is the genotype in this case. That is just the types of genes these parents have. And those genes lead to certain phenotypes, which is the color of the fur that these mice have or the hair that they have. And those are the phenotypes. So in prokaryotes, we usually don't have anything quite as obvious as this. So with prokaryotes, we can't do Punnett square crosses, and we usually don't have really obvious physical characteristics to observe for the phenotypes. We're more looking at the metabolic characteristics of the organism. However, this is a really good visual to help you understand the difference between phenotype and genotype. Genotype, remember, is just the order of the nucleotides, the types of genes the organism has, and then the phenotype is how that organism looks once those genes are used to make proteins. So now that we've gone over some of the fundamental terms and concepts that we work with when we're talking about genetics and heredity and gene expression, etc. We're going to talk about the roles of DNA, RNA, and proteins in genetics overall. So we're going to go into more detail about each of these in the next couple of slides, but just to let you know what we're going to be talking about on those slides, we'll talk a little bit about DNA, and remember DNA is the nucleic acid that contains the code for an organism. DNA basically tells an organism what it is, what to do, and when it should do those things. RNA is a second type of nucleic acid that basically serves as an intermediate molecule between DNA and proteins. So DNA contains the information that the organism needs to use to make proteins, but when an organism needs to make proteins, it doesn't just take the information in DNA and create a protein. Instead, it goes through RNA, and then, so the information contained in DNA goes through RNA in a couple of different ways. And then the information contained in that RNA is actually used to directly make that protein. And then proteins are the macromolecule that are made as a result of the code in the DNA. So proteins are the molecules that really give cells and organisms all of their functions and their functional characteristics. I'm not saying that things like lipids and carbohydrates aren't important either. They certainly are. However, it's proteins that are actually used to assemble those types of molecules, to place those types of molecules, to make sure those molecules get used for what they need to. And of course, in addition to the important functional roles that proteins play in organisms, they also have significant structural roles, whether that role is helping support a membrane or create a cytoskeleton, or perhaps make some kind of motility structure like a flagellum or a cilium or fimbriae or something like that. So proteins play an important role in the structure of organisms in addition to the functioning of them as well. In terms of DNA, in this slide we go into a little bit more detail about the structure and function of the molecule itself. So as you probably recall from previous classes and just general life knowledge, DNA is a double helix molecule. It is a molecule that has two backbones or two strands that kind of wrap around each other. The backbone is made of sugar and phosphate molecules, and then inside that, the rungs of what you would consider this kind of twisted ladder are nitrogenous bases. Now, we often refer to these as base pairs because they pair with each other. So there are 4 types of nucleotides that are in the bases of DNA, or the nitrogenous bases of DNA. Those are A, G, C, and T. And the nitrogenous bases, the nucleotides, are the individual subunits of the DNA molecule. So basically, if you look at a long strand of DNA, that is a macromolecule, and the individual subunits, the individual things that make up that macromolecule, are the A's, G's, C's, and T's. When we're looking at how the nucleotides pair with each other, A always pairs with T, and C always pairs with G. And the way I got taught to remember that way back in high school biology was that A and T have straight lines in them, and they always pair together, and then G and C are curvy letters, or they have curved lines in them, so you can remember it that way. There are lots of different ways to remember that however you want. That was just what my teacher taught us, was that if you just remember that A and T pair together because they have the straight lines in them, and then C and G pair together because they have curved lines in them. So we've got our DNA molecule, which is this double helix molecule with these backbones containing the nitrogenous base pairs that kind of wrap around each other. And those nitrogenous bases, these A's, G's, C's, and T's, are the individual subunits. Now, the order of those individual subunits matters a lot because the order of those end up making up genes. So genes, the basic definition of a gene, is several nucleotides in a specific sequence, so not just a random sequence, that code for a protein. So when we talk about the genes of an organism, we're not just talking about random sequences of DNA. We're talking about chunks of DNA, specific orders of nucleotides found within this larger DNA molecule that code for a protein. There is a very specific meaning of the word gene, and it doesn't just mean all of the random DNA in an organism or just random sequences of DNA. It has this very specific meaning, and a gene is basically a specific order of nucleotides that will ultimately lead or can ultimately lead to the creation of a protein. Now, when we get a bunch of genes stuck together, all on a single strand or single structure of DNA, that is called a chromosome. So right now you're looking at a eukaryotic chromosome, and the chromosome is actually just one of these guys. So this is a chromosome, and right now it happens to be paired with another of the same type of chromosome. But really, when we're talking about a chromosome, at least for a eukaryote, this is an example of a single chromosome. When you see this X shape, it's because you've got two chromosomes of the same type stuck together in the center here. So in eukaryotes, most of the time there are multiple of these linear kind of X-shaped chromosomes in there. In prokaryotes, remember, there's just typically one circular chromosome. So if we have this, and then we have like our other side of our DNA, and then we would have our nitrogenous bases and their pairs inside here. So this is what a chromosome for a prokaryote would look like. Obviously not drawn to scale and not a very good artistic representation, but that's what you get. Now, when we talk about all of the genetic material in an organism, whether we're talking about all of the chromosomes in a eukaryote or the chromosome plus some plasmids in a prokaryote, we're talking about the genome. So in a eukaryote, typically the genome just consists of the chromosomes that it has. And for the most part, eukaryotic genomes are stable throughout the individual organism's lifetime. So the chromosomes that a eukaryote is born with basically are the same ones that it's going to die with, and its genome doesn't change very much throughout its life. However, with prokaryotes, the genome can be quite fluid throughout an individual's lifetime. The reason for that is most prokaryotes are going to have at least one chromosome, but they may gain or lose plasmids throughout their lifetime. So a prokaryote could start its life with a chromosome and two plasmids, and at some point it loses a plasmid, and then later it gains two plasmids, and then it loses some. So it can change for prokaryotes throughout its existence. On this slide, we're going to go into a little bit more detail about RNA structure and function. On one of our previous slides, we just talked about kind of an overview of RNA and how it can serve as an intermediary between DNA and proteins, and that is true. But here we want to go over the three types of RNA that we find in organisms and what their individual roles are. RNA is a type of nucleic acid, and most of the time we find RNA in a single-stranded arrangement. So we don't have that double helix, we don't have that twisting that we find in DNA. Occasionally we do find some double-stranded regions, but for the most part, for RNA, there is a sugar phosphate backbone, and there are nucleotides, which again are the individual subunits of this type of nucleic acid that are attached to this backbone. The individual nucleotides in RNA are a little bit different than the ones that we find in DNA. So both DNA and RNA have A, which is adenine, both have G, which is guanine, both have C, which is cytosine, but in place of T, which is thymine, RNA has U, which is uracil. So in DNA, we have A, G, C, T, but in RNA, we have A, G, C, and U. In terms of the pairing, we're not going to worry about the pairing too much, but in RNA, A would bind with U because there is no T. G would bind with C, C would bind with G, and U would bind with A. Again, it's not going to matter too much to us for what we're going to be talking about, but just in case you needed to review that, you have that information. There are 3 types of RNA that we are going to discuss here. The first is called messenger RNA, or mRNA. Messenger RNA is single-stranded, and it has nucleotides on a sugar phosphate backbone that are arranged in groups of 3. These groups of 3 nucleotides are called codons. Each codon corresponds to a specific amino acid that can be used to create a protein. rRNA is ribosomal RNA. And this type of RNA is found in the ribosomes of organisms. They help make up the structure of ribosomes, and they are important in helping identify organisms. So ribosomal RNA is very unique in that it is highly specific when you're trying to use it to identify individual types of organisms. We can take the sequence in ribosomal RNA and figure out exactly what type of organism, exactly what species, etc. that we are looking at without having to look at other types of DNA or RNA or other types of genetic sequences. So that is a very interesting and kind of unique thing about ribosomal RNA, is that it is so very specific. It is like a very, very specific identification marker to different organisms that we have been able to use that to help classify organisms, to help group them, to help look at evolutionary relationships. And in fact, it was looking at ribosomal RNA sequences that allowed organisms to be grouped into 3 domains of life. Before being able to understand RNA sequencing or ribosomal RNA sequencing, we just had basically 2 domains of life. We had the prokaryotes and the eukaryotes. But it was looking at these ribosomal RNA sequences in various prokaryotes that led to the development of the 3-domain system, because it was found that the ribosomal RNA in certain organisms, certain prokaryotes, was very different than the ribosomal RNA found in what we now call the bacteria. And the difference was so significant that it warranted separating out prokaryotes from each other and creating 2 domains from them instead of just 1 domain. So that's kind of a fun fact about ribosomal RNA. Now, our last type of RNA that we're going to talk about is the tRNA, which is called transfer RNA. And tRNA is used to carry amino acids. tRNA is one of the few types of RNA that we can find a couple of places where it's double-stranded. And one of the places where we find that, or near one of the places that we find that, is a special structure called the anticodon. The anticodon basically has the exact opposite, or the complementary code to the code that's found on a codon, on mRNA, so that when there is a codon and a complementary sequence on an anticodon, those 2 can match up and an amino acid can be placed in a protein. So anticodons are groups of 3 nucleotides on a transfer RNA, and they have the complementary sequence to what is found on a codon. So if a codon said let me switch to the pen. If a codon, an anticodon, if a codon had the sequence ACG, then an anticodon would have the sequence UGC. Now we can look at the sequence of codons and figure out which amino acid will be placed where in a protein. You guys will do a little bit of work with this, but not too much, but I still want to explain the codon chart and either review it if you remember doing this in a previous class or introduce it if you don't know it. So if we look at our codon, we look at the order of the nucleotides in the codon. We would say, we'll just use this as an example, ACG. A is our first one, so we look for our first base pair in the codon. So we go to our chart and we say, this is U, okay, that's not it, C's not it, A, okay, so we start here. Then, to find the C, which is our second base in the codon, we look up here and we say, okay, where is the column for the C? So it's here, so now we have AC. And our third base is G, so we look over here. So we're in the A row, the C column, and then we look for the G right here. So ACG codes for the amino acid called threonine. So the tRNA that basically this codon is calling for would have the anticodon UGC, and it would be carrying the amino acid that we call threonine. Don't worry too much about the codon chart right now. Don't worry if you don't know what these abbreviations mean in terms of what amino acids they correspond with. In this class, we're not going to be doing a ton of work with trying to figure out genetic sequences or anything like that. I'm just reminding you of how this all works or introducing it at least briefly if you didn't get a good grasp of that during your introductory biology class. But in general, the big stuff to pay attention to on this slide are the 3 types of mRNA, their basic functions, the basic components of them. So we have messenger RNA, ribosomal RNA, transfer RNA. Messenger RNA is arranged in codons, and it will meet up with transfer RNA. The codon on the messenger RNA will attach to the anticodon on the transfer RNA, and this is what allows individual amino acids to be placed in proteins. We'll talk about all of that more in depth in the next several slides, but that's just the big picture for here. And also, RNA is really cool. It helps make up the structure of ribosomes and plays a significant role in understanding evolutionary relationships and identities of organisms. So far, we have covered DNA structure and function, RNA structure and function, and now we want to do a little bit about protein structure and function, and then we can kind of put all of that together and start to understand how information in the DNA gets turned into proteins. So proteins are macromolecules that are made up of amino acids. So the polymer is called a protein and the monomer is called an amino acid for this type of molecule. And while there are many types of amino acids that exist in the world, there are approximately 20 amino acids that are used by living organisms on a regular basis. So you have a chart of the 20 most common amino acids for organisms to use to make proteins. Every once in a while, there will be another one slipped in here periodically, but by and large, these 20 amino acids are the 20 amino acids that all organisms on the planet use to make up proteins, no matter what type of organism they are, whether they are a bacteria, an archaea, a plant, an animal, a fungus, all of them basically use the same 20 amino acids to make up proteins. You don't need to worry about memorizing their names, their shapes, their chemical compositions, or anything like that. One thing I do want you to pay attention to when you look at the amino acids is how each of them has kind of a core structure, this H2N and then COOH group, and then they will have an extra group on the outside, an extra just tail or ring shape or something like that. And that is significant because basically all amino acids have this core structure in them, but each of them has an extra chain or two or ring or something added to the outside of that core structure. And it's that extra part of the molecule that allows proteins to have so many different shapes and so many different functions. If all we had was a bunch of these base parts of the amino acid being joined together, then we wouldn't really have any diversity in terms of protein structure or protein function. So if all we had was a bunch of these little H2N COOH groups being joined together, every protein would be exactly the same. However, because each amino acid has its own extra group on the outside, and I'm just highlighting some of them, not every single one of them, but because every amino acid has an extra chain or extra ring or extra atoms added to it on the outside, that is what allows organisms to have the ability to create so many different types of proteins, so many variations of proteins. It basically is because of these extra groups that are found on the core structure of amino acids that allows for this incredible diversity of protein structure and function. Each protein that an organism produces or contains in its body has up to 4 levels of organization. So as a whole, proteins, this group of macromolecules that are made up of amino acids, have up to 4 levels of organization. The first level of organization is called the primary level of organization, or creates what we call the primary structure of the protein. And this level is made up of just the amino acids and the order that they're in. So we have 20 different amino acids, and the order that those are put in to what we call the polypeptide chain makes up the primary structure of a protein. Not every protein is going to contain all 20 amino acids. Sometimes they may have 5 or 10 amino acids that are just repeated frequently in that, what we call the polypeptide chain, or that primary structure of protein organization. Sometimes they may have 15, they might have all 20 common amino acids in the primary structure. But whatever combination of amino acids they have, the order that they are put in is what makes up that primary structure. The secondary structure causes when the primary structure essentially folds or twists around itself. So there are 2 types of structures that can be found in the secondary structure of a protein, or the secondary level of organization. And that is either an alpha helix or a beta pleated sheet. So oftentimes this one will be called an alpha helix, just like it says here. This one will sometimes be shortened or abbreviated to a beta sheet. Its technical name is beta pleated sheet, but sometimes it's abbreviated to just beta sheet. So after we have just the order of amino acids, those amino acids start to interact with each other, and they cause the secondary structure to form. And that secondary structure means the amino acids are either twisting around themselves and creating this alpha helix, or kind of folding up like a piece of paper and causing this beta pleated sheet to form. Next, we have the tertiary structure. This is caused by the amino acids interacting with each other even more, and this creates the overall shape of a protein. When we're talking about proteins, there are kind of 2 very broad categories, and those are kind of the linear proteins, which are fairly straight overall. They will have regions maybe of alpha helixes, regions of beta pleated sheets, but overall the protein itself is more straight in nature. And then you also have what's called the globular proteins, which are like globs. They're a lot more round. So when we have a globular protein, instead of the overall shape of the protein being rather straight, it kind of curves around itself and creates a ball-like structure. Almost never would you find an actual perfect sphere when we're talking about protein structure, but you get the idea that basically the amino acids are interacting with each other and kind of curling around themselves to create a spherical structure, which is kind of ball-like in nature. Now, many proteins are done once they reach their tertiary level of organization. They're ready to go, they're ready to function in the organism, ready to perform their task or their job, whatever that is. But other proteins have to have one final level of organization, And this is called the quaternary structure, or quaternary. It depends on how you pronounce it. Either way is fine. For this type of protein to form, what happens is we have two separate tertiary structures come together. So basically what we have is two individual proteins join together to make a larger protein complex. Now, like I said, not all proteins have this. Many proteins are functional. They're ready to go at their tertiary level of organization. But some proteins, to be able to function the way they need to, actually have to combine with another protein to make this fourth level of organization. So now, at this point in the lecture, we have talked about DNA structure and function. We've talked about RNA structure and function. We've talked about protein structure and function. And it is time to combine all of that information and talk about transcription and translation. Transcription and translation are the processes that cells use to take the information contained in a gene, or take the information contained in a DNA molecule, and turn that into something functional, which happens to be the protein. So if you're familiar at least a little bit with the concept of 3D printing, basically DNA would be the instructions in the machine, and the protein would be the thing that gets created with those instructions that you put into the machine. Together, the processes of transcription and translation are referred to as gene expression. So when you hear the term gene expression, what that means is taking the code in DNA and creating proteins from it, and that happens in two steps. It happens in transcription and translation. Now, transcription and translation each have three of their own steps, and we're going to look at those steps on the diagram that I have here on the slide. So on the left-hand side of this diagram, the process of transcription is occurring. On the right-hand side of the slide, the process of translation is occurring. Now, each of these processes, transcription and translation, like I said, have three phases. Each have an initiation phase, each have an elongation phase, and each have a termination phase. So we'll start with transcription and talk about initiation, elongation, and termination in transcription. In transcription, what happens is that the code in DNA is used to create a molecule of messenger RNA, or mRNA. And transcription initiation begins when a molecule called RNA polymerase, which is represented by this blue structure here. I'm just going to write that out for you so you know what to label. So initiation of transcription begins when RNA polymerase finds the beginning of a gene in a DNA molecule. So basically this RNA polymerase, which is a big protein complex, runs along DNA until it locates the beginning of a gene, until it finds like, oh, hey, this is where this gene starts. This is where I'm going to start things. Once it finds the beginning of a gene, it causes the backbones of the DNA helix to separate a little bit from each other. So those backbones separate a little bit from each other, so the nucleotides are exposed, those nitrogenous bases, the A's, G's, C's, and T's. And that allows RNA polymerase to copy the code that's in DNA and start creating mRNA. Elongation of transcription is essentially just the process of RNA polymerase running through the DNA, copying the code, and creating the mRNA, making that strand of mRNA longer and longer and longer until that mRNA reaches the end of the gene, and then termination will occur. So termination of transcription occurs when RNA polymerase finds the end of the gene, it finds a stopping point, and the full molecule of mRNA has been created. So here we've got the mRNA molecule in dark blue, basically, and RNA polymerase has said, okay, I've reached the end of the gene. I'm not going to copy any more code from the DNA. So I'm just going to label some things really quick for you. This is mRNA. And this is also mRNA. And then hopefully yellow will show up. Basically, this just goes time. So time happens. This is time point 1. This is time point 3. So just again to recap, transcription is a process of taking the code in DNA and using that to create mRNA. It occurs in 3 phases: initiation, elongation, and termination. Initiation of transcription begins when RNA polymerase finds the beginning of a gene on DNA, causes the backbones to separate, and it starts to copy the code in DNA and starts to create mRNA. Elongation of transcription occurs as RNA polymerase is copying the code in the DNA and creating mRNA and just making that mRNA strand longer and longer. Termination of transcription occurs when RNA polymerase reaches the end of the gene in the DNA and no more A's, G's, C's, or U's are added to the mRNA strand. The mRNA strand is basically ready to go on to the next phase. So the next phase is translation. And translation is a process of taking mRNA and creating proteins. This process also has initiation, elongation, and termination. And initiation occurs when mRNA goes to the ribosomes. In this diagram, the ribosomes are the gray structures. And if you think back to Unit 1, remember talking a little bit about ribosomes. Ribosomes are protein RNA complexes. They have a big and a small subunit. And in between the big and small subunit, there is a space. And in this space, mRNA can insert itself. So initiation of translation begins when mRNA reaches a ribosome and it has inserted itself into the space between the big and small subunit. Once the mRNA is there, the transfer RNAs or the tRNAs come over and start looking for their corresponding regions on the mRNA. So remember, mRNA is arranged in codons and tRNA has anticodons. And so during this process, the anticodon of the tRNA is essentially looking to figure out which codon of the mRNA it can match up to. As the tRNAs find matches between their anticodon and the mRNA codon, then the amino acids that the tRNAs are carrying begin to join together. So this process is called elongation. And what's happening here is that the beginning stages of a protein are forming. When we have tRNAs who have found the right match between their anticodon and the codon on mRNA, and they're just sitting together like this in the ribosome, then the amino acids that are attached to those tRNAs will bond to each other through what are called peptide bonds. That's a P, peptide, not some weird letter that doesn't even exist in English. So these are peptide bonds, and they form when amino acids that were attached to transfer RNAs are sitting next to each other and they attach to each other. Once those amino acids attach to each other, they start to separate from their tRNAs. So elongation of translation is just this repeated process of tRNAs that have these anticodons and amino acids coming over and finding out whether they can join to a codon in the mRNA strand. And if they can, then essentially what happens is the tRNA comes in, sits in the ribosome, the amino acid that was attached to that tRNA gets transferred to this first part of the protein that's forming. This first part of the protein is called a polypeptide chain. And so this process just happens over and over and over until the next phase, which is termination, starts to occur. Termination occurs when there is a codon in mRNA that has no corresponding anticodon in tRNA. So at the end of every mRNA strand, there's basically a codon that has no match to an anticodon on tRNA. Once that codon at the end of mRNA runs through the ribosome, there's no way to string any more amino acids on this polypeptide chain because there's just no tRNA that would carry an amino acid and have its anticodon match up with the codon here. So the polypeptide chain is done. It is released from the final tRNA, and the mRNA can then either be used to create another copy of this polypeptide chain. The tRNAs will be recycled, more amino acids will be put on them, and they can be used again to create more proteins. And this polypeptide chain goes on to start going through its levels of organization and forming a protein. So at this point, this is basically kind of the primary level of protein organization. We have what we call our polypeptide chain. And then when those individual amino acids when those individual amino acids start to interact with each other, that will start causing either the alpha helix to form or the beta pleated sheet, and that will cause a tertiary structure to form, and potentially that will lead to that protein joining with another and creating a quaternary structure. On this slide, you just have a close-up view of the diagram from the previous slide. That way, if you want to print it out or you want to put it on a tablet or something and mark it up more and just look at it in more detail, you can do that. So just remember, this side is transcription. This side is translation. And with transcription, we are taking the code in DNA and creating mRNA. With translation, we are taking the mRNA and using it to create a polypeptide or a protein. Each of them has 3 phases, which is initiation, elongation, and termination. Before we finish up our discussion on transcription and translation, or gene expression, I just want to do a little bit of comparing and contrasting these processes in prokaryotes and eukaryotes. Overall, the processes of transcription and translation are incredibly similar in prokaryotes and eukaryotes. In both cases, the organisms go through transcription, where they copy the code in DNA and create mRNA. And in both cases, they go through translation, where they take that code in mRNA, put it into a ribosome, and essentially out comes a protein. So in that respect, they are incredibly similar, if not identical. But there are some small differences in terms of how this works at a molecular level. So for prokaryotes, because they have no nucleus, the process of transcription and translation is essentially continuous or simultaneous, and it happens in the cytoplasm. So if you think back again to Unit 1 and you think about that structure of a prokaryotic cell, they have the nucleoid region where their DNA is, and basically right next to that, you will find the ribosomes that are needed to be able to create proteins. So transcription and translation can happen one right after another, virtually simultaneously. Of course, there's a little bit of time that it takes to create the mRNA and get it read in the ribosomes. But basically, once a prokaryote is able to create mRNA, a protein can be made immediately after that. And so the process of protein creation is much, much faster in prokaryotes than it can be in eukaryotes. The reason for that is in eukaryotes, transcription and translation are a little bit physically separated from each other. In eukaryotes, transcription takes place in the nucleus. So remember, eukaryotes have a nucleus that contains all of their DNA, and RNA polymerase has to essentially travel to that nucleus, read the DNA, create the mRNA, and then the mRNA has to be sent out of the nucleus into the cytoplasm of the cell, typically to specifically the endoplasmic reticulum, so that the proteins can be made, or so that translation can occur. So in eukaryotes, transcription occurs in the nucleus, And translation occurs in the cytoplasm, usually around the endoplasmic reticulum. Another interesting thing about transcription and translation in prokaryotes versus eukaryotes is that prokaryotes, when they make mRNA, it is ready to go. As soon as RNA polymerase makes mRNA, it can be used directly as it is to make a protein. In eukaryotes, oftentimes, if not the majority of times, the mRNA structure is a little bit different. So the mRNA structure is still just a single strand of nucleotides strung together, arranged in codons, but in eukaryotes, they also have regions of that mRNA called exons and introns. Exons actually are regions of mRNA that will match up with amino acids. Introns, on the other hand, are seemingly kind of random regions of mRNA that won't match up with tRNAs. So mRNA in the eukaryote has to be processed in that the eukaryote has to take out the introns and put all the exons together before it can be sent to a ribosome and used to create a protein. So this is what eukaryotic mRNA looks like once it just comes off RNA polymerase. It's got regions that are going to be able to be used to create a protein, and then regions that are not going to be able to be used to create a protein. Those introns have to be cut out, essentially. So the eukaryote has to get rid of these regions and then splice all of the exons together. So this is called pre-mRNA, and we've got introns and exons. And those introns have to be taken out and the exons have to be put together so that the mature mRNA can be created. And this will then be able to be sent to the ribosome to create a protein. If those introns are not taken off, then the protein can't be created. Now, why these are there, we're still not 100% sure of. However, we have figured out if those introns are not in the mRNA, at least initially, everything kind of gets garbled. Like the message doesn't go through the way it's supposed to. So the introns are needed. We're not exactly sure for what, but we know that if they are removed at the DNA level, then the message gets garbled and the mature mRNA does not have the right structure and does not create the right type of protein in it. So don't worry too much about that aspect. I just know sometimes people have questions about it. But what you want to remember is that from this slide, basically, for the most part, the process of transcription translation in prokaryotes and eukaryotes is the same. There are some differences. Prokaryotes is continuous and it all happens in the cytoplasm. Whereas in eukaryotes, transcription and translation have to take part in separate locations. So transcription takes place in the nucleus, the pre-mRNA has to be processed, the introns have to be removed, the exons have to be spliced together or put together, and then once that mature mRNA is ready, it can be sent for translation in the cytoplasm, which typically happens at the endoplasmic reticulum. In our last several slides, we talked about how proteins are created. So we talked about the process of gene expression. We talked about the two steps in gene expression, which are transcription and translation, and went into details about exactly what happens in each of those stages. And now we are going to talk a little bit about how prokaryotes decide when transcription and translation is going to occur. So on these next few slides, I am just going to be talking about prokaryotes. Eukaryotic gene expression is totally different than this. So just remember that on these next couple of slides, I am just talking about prokaryotes and really focusing on how prokaryotes determine when transcription and translation should take place and what types of proteins they should produce when. As you know, gene expression is the process of producing proteins from the code in DNA. And most organisms are capable of producing thousands of different types of proteins. However, not every one of those proteins is needed at every moment in time. And in fact, sometimes producing every single protein that they're capable of would be really problematic because one, it would use up a bunch of energy. Two, it would require a lot of nutrients and a lot of raw materials. And three, that organism may create proteins that actually compete with each other. So let's say an organism can produce proteins A through Z, and proteins B and protein L have completely opposite functions of each other. If that organism produces protein B and protein L at the same time, those two proteins are going to interfere with each other, and the organism will be inefficient at best and dead at worst. To avoid this situation where an organism is producing every single protein it's capable of and potentially killing itself in the process, they use a process called gene regulation. Genes and gene expression are regulated so that proteins are produced at ideal times. Basically, this gives the organism the ability to pick and choose which proteins are produced when, generally in response to environmental conditions, so that the organism produces the right proteins for that moment in time, and it can be as efficient as possible. In prokaryotes, genes are organized into structures we call operons. And operons are kind of like light switches. They allow the organism to either turn on genes or turn off genes. If a gene is turned on, then proteins will be produced. If a gene is turned off, then those proteins will not be produced. Operons themselves are regulated by repressors, and repressors are molecules that keep proteins from being made. Oftentimes, repressors are proteins, not necessarily always, but most of the time, repressors are going to be proteins. And what these repressors do is interfere with RNA polymerase's ability to attach to the gene and create mRNA. So an operon is essentially a structure or a complex that controls whether a certain gene or group of genes are expressed or not. There are three main parts to an operon. So what you're looking at here in this diagram is an operon, and this is our prokaryotic cell. We find operons literally just in the code of DNA. So if you were to magnify DNA and be able to zoom in a lot and understand what all of the nucleotides meant in a specific order, you would find that the orders of certain nucleotides were essentially creating operons within the DNA. So back to these three main parts. The three main parts of operons are promoters, operators, and structural genes. Promoters are the places on operons, or the places on the code in DNA, where the RNA polymerase binds. This is when we were talking about kind of initiation with transcription translation. This is where initiation would occur, at the promoter region. The operator is a place on the operon where a repressor can bind. Now, like I said, most of the time repressors are going to be proteins. Maybe not always, they could be other things, but most of them are proteinaceous in nature. And so a repressor will literally attach to or kind of encircle this region of DNA to potentially block RNA polymerase from moving any further. And then we have the structural genes. The structural genes are actually the genes that the organism would use to create a protein. So these are the genes that are used to create proteins. These are the regions of DNA that actually will code for a functional product, which is the protein. So if we look at our operon here, we see our three regions. We have our promoter, our operator, and our structural genes. I'm going to go ahead and just label some things so you remember what they are. The promoter, that's the region where the RNA polymerase would attach to and go through initiation for gene expression. This is RNA polymerase. The operator is where a repressor would sit. And then these are the structural genes, and they code for proteins. If we have a repressor sitting on the operator, then even if RNA polymerase comes over and finds the initiation site for transcription, it will start to try to move forward, but it can't. The repressor is blocking it, so the RNA polymerase can't reach the structural genes. If RNA polymerase can't reach the structural genes, then mRNA cannot be created, and if mRNA cannot be created, then a protein cannot be created. So that is the basic structure of operons and how they essentially work. If the repressor is there, RNA polymerase can't reach the structural genes and can't create proteins as a result. However, if the repressor is gone, then the RNA polymerase is free to move along this DNA molecule, reach those structural genes, create mRNA that can be used to create a protein. Prokaryotes have two types of operons. They have operons that are basically always switched on unless something in the environment tells them to turn off. And they have operons that are switched off until something in the environment tells them to switch on. We're going to talk about those two different types on the next two slides, which also happen to be the last two slides of the lecture, so you're getting close to being done. Inducible genes are genes that are controlled by operons that are normally in the off position, which means that most of the time, the organism is not using these genes to create proteins. So most of the time with inducible genes, the repressor is sitting on the operator, and even if RNA polymerase happens to be bound to the promoter, the repressor will block its movement, and so the RNA polymerase cannot reach the structural genes and no proteins can be made. However, if something in the environment binds to the repressor and causes that repressor to change shape so it falls off the operator, then the RNA polymerase is going to be able to reach the structural genes and proteins will be able to be made. This specific example that you're looking at here on this slide is called the lac operon. Now, there are lots of different inducible genes out there. There are lots of different operators with different names. This is just one example. It's kind of the classic example of how inducible genes can work in bacteria. So please don't think this is the only type of inducible operon there is. That is not true. This is just one example, and it's kind of the classic example because it's very easy to understand and was kind of one of the first ones discovered. So in the lac operon, this operon structure, this genetic structure, contains the genes that are needed to make an enzyme called lactase. Lactase is an enzyme that will break down the sugar called lactose. So lactose is what we call a disaccharide, meaning it has two sugar molecules joined together through a bond, a glycosidic bond. And this is lactose right here. Now, lactose is not necessarily a common sugar to find in the environment. It is not found in most environments where many bacteria are. However, if a bacterium happens to be placed into an environment where lactose is available, then lactose can be a very good source of energy to make ATP. So, for a bacterium that has the genes to make this enzyme called lactase, most of the time they are switched off because most of the time the bacterium does not have lactose available to it. Most of the time these bacteria are in an environment where this sugar is not present, and if it were to make this lactase, this enzyme, when lactose is not present, then that's a waste of energy and a waste of raw materials. However, if lactose is present, then it is very useful to create this enzyme called lactase because lactose is a good source of energy to make ATP. So most of the time, the repressor for this operon is bound to the operator. And that means the genes that are needed to make lactase are not being expressed. So what this means in this situation, this is kind of the normal situation for these bacteria. The repressor is bound. RNA polymerase cannot move to the structural genes that are needed to make lactase. So lactase is not getting made. But if lactose is suddenly in the environment for whatever reason, there are lots of reasons that can happen, then lactose itself, the sugar molecule, will actually bind to the repressor here. The binding of lactose to this repressor causes that repressor to change shape. As a result, the repressor falls off, and now the RNA polymerase can move forward and can reach these structural genes. So proteins can be made. Once those proteins are made, once the RNA polymerase is able to make mRNA, and the mRNA is able to go through translation and make those proteins that these genes code for, the enzyme called lactase is created, and the bacterium has the ability to break down this sugar and use it to make ATP. So again, this is just an example. This is kind of the classic example of inducible genes or an inducible gene. And remember, with inducible genes, they are switched off until something in the environment causes them to switch on. In this case, that something in the environment would be lactose, a potential source of energy. Most of the time, this operon is switched off because it would be pointless to make lactase if there's no lactose available. But if lactose is available, lactose can bind to the repressor, cause it to fall off. The RNA polymerase will move forward, it will reach the structural genes, and it will create lactase, which is the enzyme that is needed to break down lactose, and the bacterium now has an additional source of energy to create ATP. Now, the other type of operon is called a repressible operon. These are repressible genes. And these are genes that are normally switched on because their products, or the proteins that are created from these genes, are needed regularly by the bacteria. So in this case, with these types of genes, they are switched on most of the time because they create a protein that the bacteria needs in almost all environments all the time. They only turn off when some kind of environmental condition tells them, hey, stop making these types of proteins because we don't need them anymore. Our example for this type of operon is called the TRP operon, or the TRP operon. This operon is responsible for making proteins that will allow the bacterium to manufacture something called tryptophan. And tryptophan is one of the 20 amino acids that are normally used by organisms to make proteins. So for most bacteria, they need to be making proteins constantly, and so they need a constant supply of amino acids, and one of those amino acids is tryptophan. So what we have with the TRP operon is that it is turned on almost all of the time. What we have is we have our promoter, we have our operator, there's no repressor on the operator is basically free, and so the RNA polymerase is constantly being able to reach the structural genes. In this diagram, the structural genes are in this dark purple color. And when these genes are expressed, or these genes are actually, the information in them is turned into mRNA, and that's used to create proteins, then that creates an enzyme that will put together tryptophan. So most of the time, this is on all the time because bacteria always need tryptophan, and therefore they always need the enzyme that will be created by this operon because they always need to manufacture tryptophan so they can make more proteins. However, when their growth slows down for whatever reason and not as much tryptophan is needed, then tryptophan will start to build up in the cell, and the tryptophan will actually attach to the repressor, which will activate it. It causes the repressor to change shape, and it will be able to bind the operator. Once the repressor binds the operator, RNA polymerase can no longer reach the structural genes. It kind of gets stuck behind that repressor. Those structural genes cannot be reached anymore. Therefore, gene expression cannot occur. The organism cannot make the enzyme that is needed to make tryptophan, and no more tryptophan is produced. Now, this can occur for many different reasons, just like most things in the world of microbiology. But one good example would be if a bacterium were going to be going into an endospore form and its growth was slowing down, it wouldn't need as much tryptophan anymore because it would be focusing its efforts on creating that endospore and not actively going through all of this protein production. So don't take that to mean that only bacteria that produce endospores have this operon. That's not true. That's not what I'm saying. This is just an example of when this operon might be used to stop the production of proteins.